Hi, thank you so much for staying for my presentation. My name is Aixa Andrade, and as he said, I come from UT Southwestern Medical Center. Um, so let's talk about single cell RNA-seq data. These data take samples from a tissue and then they are sequenced to get a single cell gene expression profile. And what I analyze is a count matrix of N cells by M genes. And some characteristics of this data is that it is a very high dimensional data. It's very sparse, and sometimes the data is confounded. Those confounders are also known as batch effects. And when it's confounded and you reduce the dimensions, sometimes it looks like this. This is a two-dimensional latent space with batch effects in which the cells are clustered by batch, which are the colors instead of by cell type. Some sources of batch effects can be technical, as you may know, like library preparation or different experiments, even different labs or sites. And they can also be biological, like samples coming from different donors. And we are interested in those kind of effects that are also biological. Some options that you have for batch effects correction is to remove the batch effects to have a batch invariant latent space, like the blue one that we have down there, in which the cells cluster by cell type instead of by batch, or to model them to account for batch variability. So we like the second option because we think that some batch effect correction methods remove important biological information, and they may even remove important donor information. So let's talk about existing methods for single cell RNA-seq modeling. So as I mentioned, it's a very high dimensional data. So we use methods for dimensionality reduction. It could be PCA, which is linear. It could be Autoencoder, which is nonlinear. And there are many methods that are based on autoencoders, and they have different similarity constraints. And another popular method is to combine the autoencoders with generative adversarial networks. And in this case, they have an adversarial network, which is penalizing the model for predicting batch. And this is how you correct them for batch effects. Some limitations are that most batch correction methods act like a fixed effects model, focusing on preserved cell type signal, but they remove batch variability. And models that do learn batch distributions don't clearly separate batch specific from batch invariant effects, which makes them less interpretable. And this lack of interpretability limits their application, missing opportunities in personalized medicine. So our solution is inspired on linear mixed effects, which separates fixed effects from random effects. The fixed effects are cluster invariant and the random effects are cluster specific. So what we do is to build an architecture based on two autoencoders of networks that are trained separately. On the left, we have the fixed effects of network, which is batch invariant. And on the right, we have the random effects of network, which is batch specific. And the bottleneck is our latent space. We have two, one that comes from the fixed effects of network and one that comes from the random effects of network. And we can use these two latent spaces to combine them and obtain a mixed effects prediction of a target that you are interested. And the advantages are that we have improved interpretability. We model both fixed effects and random effects, and we also improve downstream classification accuracy. So the two experiments that I'm gonna show you are about showing the advantages of these two latent spaces. So the first one is about characterization of the two latent spaces. We want to evaluate cell type and batch separability. And for the fixed effects subnetwork, we want the cells to cluster by the cell type. And in the random effects subnetwork, we want the cells to cluster by batch to demonstrate that our network is learning the batch variability. So to evaluate the cell type and batch variability, we use the average silhouette width, which is a score that is bound between -1 and +1. And the higher the score, the better clustering. So we want to have a high silhouette score for cell type in the fixed effects latent space and a high score for random effects in the random effects latent space. Random effects or batch effects. And the second experiment that I'm gonna show you is answers to the question, does the latent space combination improve diagnosis? So as I mentioned, one subnetwork discards batch variability and the other one models that batch variability. So they are both complementary, and we want to know if using the combined information, we can get better prediction accuracy. So we have two data sets. The first one is the healthy heart data set. We chose this data set because it has a high number of batches, in total 147, and the batches are a combination of different protocols, tissues, and donor samples. And we also have the ASD data set in which we identify the batch effects as different donors, and we have 31 donors. We have samples from ASD and controls. And the results for experiment 1 in the healthy heart data set are shown here. In the first row, we have cells colored by cell type. In the second row, we have cells colored by batch. And in the first column, we have the UMAP spaces for PCA, in the second column for the autoencoder, in the third column for our fixed effects subnetwork, and in the fourth column for our random effects subnetwork. So we can see here that our random effects subnetwork is modeling the batch variability because the cells no longer cluster by cell type, they cluster by batch. And we can also see that our fixed effects latent space is batch invariant because we have a better batch mixing than in the PCA and the autoencoder. We can also see that the UMAPs agree with the average silhouette score for both the random effects, which is now positive in the batch section, and in the fixed effects, which is more negative for the batch section as well. And something interesting that we can see here is that we have some cells that were not assigned any cell type, which are circled in red there, and they are colored in black. So in the PCA and in the autoencoder, you can see them clustering, which is a false clustering because they were not assigned any cell type. And we cannot see that big of a clustering in our fixed effects subnetwork. So we are modeling a batch invariant latent space. And now we have the ASD dataset in which we model the batches as donors. And as well, we have in the first row the cells colored by cell type, in the second one colored by batch, and our second column is the autoencoder, the third one the fixed effects subnetwork. And in the autoencoder, UMAP colored by cell type, where you can see those arrows. Those cell types indicate cells that are differentially expressed between autistic uncontrolled subjects. So you can not longer see those cells clustering in our fixed effects of network. So we model a batch invariant latent space. And some cell types that are donor invariant, we can still see them not dispersed in our fixed effects of network. So since we are removing the donor effects from the fixed effects latent space, we are also modeling them in the batch effects latent space or random effects latent space, in which you can see the cells that now they cluster by donor. And this is supported by the average silhouette scores by batch, which are higher for the random effects of network. So we have these two latent spaces that provide complementary information. And the experiment too is about using this combined information and see if it provides a better classification score. So first we classify the cell types and we obtain an improved performance. But when we use our latent spaces, actually we use a random forest classifier on the two latent spaces combined. And when we try to classify diagnoses, we get a much better score than only using PCA or the fixed effects of network than using the combined experiment. We get a much better accuracy than when using PCA or fixed effects alone. And this is all. Do you have any questions?
Very nice talk. So can you comment on, so one of the challenges, when we're doing batch effect or some kind of integration is when there's some actual biologically meaningful differences between, say, the samples and the individuals. And how have you encountered or deal with that challenge, that you don't want to adjust away potential biological differences?
Yes, so I guess that some of the challenges is removing too much batch information. Like that's the overall challenge that we are trying to target. And then we have these random effects of network that instead of just removing the batch information, models it because we believe that is useful. But one challenge that we may say is that we may have different sources of batch effects in a single data set. So to account for each one of them, using our method, you would have to have one subnetwork for each different batch effect, for example. Does that answer your question?
Thank you.
Good presentation. Have you compared your method with well-known batch correction method like Harmony or Anchor method?
And this is something that I'm working on. I don't have results to show right now. The fixed effects of network, my takeaway from all these is that since I'm training both subnetworks separately, even though if I get better results with other batch correction method by comparing with my fixed effects of network, I could just add the random effects of network as a different latent space and still use it combined with any other batch correction methods. I know that there are thousands of methods out there, so it's difficult that my method is gonna outperform every single one of them. But what I can say is that it's complementary and it provides value by also modeling the batch effects.